Welcome to Applied Photophysics Higher Order Structure video series. In this fifth and final video, we will focus on the operation of qBiC, Applied Photophysics Higher Order Structure Comparison software, via stepwise analysis of an example dataset that is included as a teaching aid with every copy of the software. If you haven't already, check out our first 4 videos in the HOS series, in which we took a behind-the-scenes look at the math underpinning HOS comparisons, reviewed a few exemplary HOS case studies, discussed practical considerations for performing HOS comparisons, and got a first glimpse of qBiC and the comparability tests it can perform. Now let's dive in. As discussed in video 4, the qBiC HOS comparison software is a program designed to perform comparability tests on sets of two-dimensional spectra. The comparison software requires data in .txt, .csv, .dsa, .dsx, or .jdx format, with the first column as wavelength and the second as CD or other measured variables such as optical rotation, fluorescence emission, or absorbance. For the .csv files, there can be as many columns as you have samples. The automation software included with our QIRScan Q100 automatically averages repeat scans, subtracts the appropriate buffer blanks, and processes data into the correct format for seamless import into the qBiC analysis software. However, when using a manual system, this data processing must be performed manually in advance before importing into qBiC. To make it easier to compare multiple datasets, the reference spectrum and sample spectra do not need to have the same number of data points, wavelength range, or wavelength intervals. These properties are not matched, the software will simply truncate and interpolate. The software also does not require that the bandwidth or time per point of the reference spectrum and sample spectra are the same, but remember that if they are different, the comparisons may not be appropriate. In the example we will discuss in this video, there are 4 datasets consisting of 12 CD spectra in 3 sets, 11 in the 4th, stored in the folder titled Protein Example Data that comes packaged with the qBiC software. The 11 or 12 spectra were acquired from separate replicates, in other words, separate cell loadings, and each is the result of averaging 3 repeat spectra, that is, 3 sequential scans acquired on the same cell loading for each replicate. The data have been corrected by subtraction of the buffer CD spectrum and exported in .csv format. The con dataset is the control, consisting of data for a wild-type protein. The 2.5% abbreviated dataset consists of the wild type with 2.5% of a mutant spiked in, and the 5% and 10% datasets consist of the wild type with 5% and 10% of the mutant spiked in, respectively. The files for replicates are numbered R1 to R11 or R12. The data acquisition parameters are summarized in the second table. Data were acquired in the far UV range from 180 nanometers to 260 nanometers using a 0.5 millimeter path length cell. The example data was easily and neatly exported in .csv format, a portion of which is shown here, and is ready for direct import into the qBiC software. The spreadsheet lists CD response values organized by sample name and wavelength. Data processing, export, and import are seamless with Applied Photophysics suite of software tools. Let's begin the operational walkthrough of the qBiC software to start analyzing these data. The first step is to create a reference spectrum. As a reminder, reference spectra should be generated from a minimum of 3 spectra collected under the same conditions as those used for the sample spectra. On the comparison software front page, click the upper graph icon, circled here, to open the spectral comparison page. And on the Spectrum menu, select New Reference Spectrum. Select the files to be used to create the reference spectrum by clicking Add Files, name the reference spectrum, and click Next. Here we have selected the .csv spreadsheet we just saw a few moments ago, which contains all our spectra, and have named the reference spectrum Control. If the selected file contains multiple spectra, then the Select Data page shown here will appear. Configure the reference selection as required by selecting the required spectra. Here we have selected spectra con R1 to con R12 to form the reference set. Note that the Previous File and Next File buttons at the top of the page can be used to navigate between multiple files that have been loaded, and Shift plus Click can be used to quickly select a range of contiguous spectra. Click Next and then Finish. The reference spectrum, which is the average of those used for the reference set, can be viewed on the Spectra tab shown here. The gray band enveloping the spectrum represents the distribution in the data, calculated as 3 sigma, where sigma is the standard deviation. The spectrum can be expanded, and the expanded area can be moved using the mouse buttons, and can be reset by double-clicking. Statistics of the reference spectrum are viewed on the Reference Stats tab. The z-score for each component of the reference spectrum is determined by comparing against the remaining 11 spectra in the reference set. Results are shown for the Pearson correlation coefficient, weighted spectral difference, and area of overlap models. The contiguous curve in the graph to the right is a Gaussian distribution based on the mean and standard deviation of the reference data. The bar chart is the actual distribution of the similarity scores, with the values being allocated to conveniently sized bins. The larger the data set, the more likely the results are to graphically conform to a Gaussian bell curve. Now that a reference data set has been established, it is time to compare the sample spectra to it. To compare spectra with a reference, select Add Spectrum for Comparison on the Spectrum menu. You will be presented with the option of either the student's t-test or the z-score. As discussed in Video 4, the student's t-test is used to compare a particular group of spectra against a reference group, whereas the z-score is used to compare a single spectrum with a reference group. If the student's t-test is selected, all files are processed and the results combined to give a single p-value. If the z-score is selected, then all sample spectra will be handled separately to give the equivalent number of similarity and z-scores. If the data file contains multiple traces, an additional page will be launched that allows the selection or omission of specific traces within the file, as we saw with selecting the reference traces. Here we are selecting the 12.5% spiked sample spectra. The overlaid reference and sample spectra can be viewed on the Spectra tab. A second spectrum is added to the spectrum view, consisting of the average of the 12 spectra used for the comparison. The significance box at the bottom of the figure shows the difference spectrum, that is, the sample spectrum minus the reference spectrum, divided by the standard deviation of the reference. Identical samples would be expected to show only a random distribution of data around 0 without systematic excursions. For example, the difference spectrum shown here has a small negative excursion between about 193 nanometers and 215 nanometers, suggesting that there are slight differences between the spectra. These differences are quantified by the statistics in the t-test tab. The t-test tab shows the similarity scores and p-values of the comparison. In this example, results are shown for the Pearson correlation coefficient, weighted spectral difference, and area of overlap models. Remember that the correlation coefficient and area of overlap models return higher similarity scores for closer similarity, whereas the spectral difference and weighted spectral difference models return lower values. For the p-values, the higher the value, the greater the probability that the null hypothesis is true, or that the two sets of spectra are the same, regardless of which model is used. The common significance level, p equals 0.05, means that a sample group with a value of p less than 0.05 is statistically different. In this example, the p-values indicate a statistically significant difference across all three similarity models. A graphical representation is shown below the similarity scores. The vertical tabs are used to select the comparison method. The reference curve in blue is composed of the results of the comparison of each of the 12 spectra contributing to the reference with the remaining 11. The position of the peak is centered on the average score, whereas the spread is calculated from the standard deviation, assuming a Gaussian distribution. The sample curve, shown in red and labeled 2.5% spiked, is calculated similarly, but is centered on the average similarity score of sample data compared to the reference. The more closely centered the reference and sample curves are, the greater the probability of similarity. Multiple sample groups can be plotted together, but each dataset must be loaded separately. Here we can see the 2.5%, 5%, and 10% data compared with the control, with all available spectra used for each comparison. By visual inspection, the spectra appear to overlay very closely, but the math tells us they are actually quite different. Note that the similarity scores decrease for the correlation coefficient and area of overlap, but increase for the weighted spectral difference as the concentration of spiked-in mutant increases. The p-value is too low to be statistically meaningful for the 5% and 10% samples. The graph shows the comparison between the three spiked data sets using the weighted spectral difference model. As the concentration of the mutant increases, the distance between the centers of the curves increases to the point where there is absolutely no overlap visible for the 10% spiked samples. For reference, this is what plots for the Pearson correlation coefficient and area of overlap models look like, both with similarity ranges from 0 to 1. It is also worth demonstrating how one or more spectra can simply be compared with the reference dataset using the z-score. The comparison statistics can be viewed on the z-scores tab as shown here. Remember that with the z-score, the lower the value, the greater the probability of similarity. Here we use the entire set of 12 spectra from the control dataset for the reference and the first 4 spectra from each of the 2.5%, 5%, and 10% datasets for the comparison. These can be selected simultaneously. We can see the reference spectrum in each of the different spectra, as well as the z-scores for the 12 sample spectra. Note the banding of the similarity scores and the z-scores into the 3 different concentrations, and that, again as expected, the similarity scores decrease with increasing mutant concentration for the correlation coefficient and area of overlap, but increase for the weighted spectral difference. In all cases, the z-scores increase with increasing mutant concentration. Before we move on to our final topic, there are a few analysis options worth mentioning. As discussed in Video 4, several spectral similarity methods are included in the qBiC software. The Pearson correlation coefficient, weighted spectral difference, and area of overlap models are displayed by default. If other models are desired, these can be toggled on and off via the Comparison Methods tab located under Options and then Settings. Another aspect of data analysis touched on in video 4 is that some of the spectral comparison methods automatically normalize the data. This means that samples measured at different path lengths or concentrations can be directly compared. But the spectral difference and weighted spectral difference methods do not, and the spectra for the same sample measured at different concentrations or path lengths will appear as different. This can be corrected on the Normalization tab found under Options and then Settings. There are 4 options for normalization. 1. No normalization. 2. Normalization by absolute area. This method calculates the area between the curve and the x-axis using step functions centered on each wavelength, treating all spectral values as positive. No additional user input is required for this option. This approach is highly recommended and is particularly useful for far UV data that may be noisy around the absorbance peak at 190 nanometers, such as when certain excipients are present. In such cases, this normalization method can provide a more robust comparison. 3. Vector normalization. The vertical of the x-axis is adjusted so that the areas above and below the axis are the same. This option is not recommended unless the user is confident about how it should be used. And finally, 4. Normalize by concentration. The sample concentration or absorbance, and in some cases other information such as path length and number of amino acid residues, need to be input by the user. To apply the same value for any variable to all the boxes, right-click and click Apply to All. This option is viable but less commonly used due to the requirement of manually entering concentration values. If the normalization option is changed after the comparison statistics have been calculated, they will be recalculated when OK is clicked. One final aspect of data analysis involves choosing the data range. The range of wavelengths included in the analyses can be reduced on the Data Range tab, also found under Options and then Settings, using either the cursor bars or the input boxes. This can be done, for example, to exclude noisy data, low wavelength regions where absorbance exceeds 2 absorbance units, or high wavelength regions where the signal is weak and contributes minimally to the analysis. If the data range is changed after the comparison statistics have been calculated, they will be recalculated when OK is clicked. All calculated reference and sample statistics can be exported via the respective buttons found under the File menu. Clicking these buttons will open a Save dialog box to generate a .csv file containing the data. Returning now to the qBiC home screen, a secondary function of the software is to compare spectra using a calibration curve. 2 data sets are used as references, referred to as samples A and B, which are different in some way. The difference may be in concentration of an impurity or degree of denaturation, for instance. In this example, we will use the wild-type protein as sample A and the wild-type with 10% mutant as sample B. We will then see where the sample containing 5% mutant lies on the calibration curve. On the comparison software front page, click the lower graph icon circled here in purple to open the calibration curve page, and on the Spectra menu, select Load Spectra. Operation is then similar to when making a comparison between spectra. You will first be asked to choose and name the file or files for sample A and then for sample B. Here we choose the 12 reference spectra for sample A, which we name as control, and the 11 10% mutant spiked spectra for sample B, which we name as 10% spiked. When both sample A and sample B have been created, you'll be asked to set the colors and number of interpolations for the calibration curve. Note that these do not affect the result of the analysis, but are only to allow you to visualize the data more easily. Click Finish to generate the calibration curve. The horizontal tabs can be used on the calibration curve page to toggle between comparison methods. Finally, input the similarity score obtained from our earlier statistical comparison. For example, we previously calculated the similarity score for the 5% mutant using the weighted spectral difference method to be 0.2376. If we input this into the similarity score box with the weighted spectral difference, we obtain a value for the fraction of protein B of 0.491, i.e. the concentration of mutant equals 0.491 times 10%, which is 4.91%, close to the expected value of 5%. We hope this 5th and final video in our higher order structure video series has been of value to you. Be sure to like and subscribe to our channel to be notified when new videos are released. For more information, including application notes, hardware and software support, and contact information, visit photophysics.com.